Okay, so the last sense that we are going to look at is our hearing and our equilibrium. And this last specialized sense is controlled by a specific organ. So just like what we did with the last senses, we will actually start off with our diagram. So if we take a look here at the diagram of the ear, because the ear controls both of these senses, the first thing that you'll notice is it's actually divided into three general sections. And these three general sections are delineated by those dotted lines. So to the left-hand side of those dotted lines is the outer ear. In between those dotted lines is the middle ear. And then to the right is the inner ear. Alright, so what we're going to do is we're going to look at the associated structures within these areas of the ear. We're going to start with the outer fleshy part of the ear, which is referred to as the auricle. Moving further inward, we have what we typically call the ear canal, and that actually has a name. It is called the external acoustic meatus. And that travels down in towards the middle ear. Once it actually reaches the middle ear, it reaches this structure, which is called the tympanic membrane. We call it the eardrum, and that's because the word tympanic actually refers to a timpani style of drum that you would see in a band or an orchestra. Attached to the tympanic membrane are the three smallest bones in the body. And the first one here has a little bulb on it, and anatomically it is called the malleus. Sometimes people will refer to this as the hammer, and the way I remember that one is because hammers are sometimes called mallets, and mallet sounds like malleus. The one that's right next to it is the incus, and that's usually called the anvil. And then the small kind of horseshoe-shaped one to the inside is the stapes, sometimes referred to as the stirrup, because if you look at the shape, it looks like a stirrup from a horse, or on a horse's saddle, I should say. Underneath the stapes is the oval window, so that is that round shape underneath there. And then beneath that is the round window. Now the other structure in the inner ear is this kind of snail shell looking thing. The main part of the snail shell is called the cochlea. But then it also has a couple of areas up here where the head would be of the snail. So this enlarged area here is called the vestibule. And then the little arches that come off of the vestibule are called the semicircular canals. So that is the anatomy of the ear that is actually involved with both hearing and equilibrium. Okay, so let's take a little bit closer look at these. So first would be in the outer ear. The function of the outer ear is to collect and direct sound waves to the middle ear. And it includes the auricle as well as the external acoustic meatus, the ear canal. Now keep in mind, I do want you to be using the anatomical terms and not the common terms for these structures. The middle ear is separated from the outer ear by the tympanic membrane, and its function is to collect, amplify, and transmit sound waves. What it does is it changes sound energy into the energy of movement, and we will see this when we go through the process of hearing. It includes the auditory ossicles, which is the collective name for the malleus, the incus, the stapes, and lots of air. Finally, the inner ear is really where a lot of the actual sensing of sound and equilibrium take place. So this is the actual sense organ itself. It includes the vestibule, the semicircular canals, and the cochlea, which taken together are referred to as the bony labyrinth. Within the inner ear, we have receptors that are called hair cells, and they communicate with neurons that are associated with them by releasing neurotransmitter. Now, these hair cells, the reason why they're called hair cells is the structure of them kind of make them look like they have hair, but they really don't. They have cilia on them, but these cilia, unlike the cilia in our respiratory system, don't move on their own. There has to be an outside force that moves them. Whatever direction these cilia moves is either going to stimulate or inhibit the release of neurotransmitter. And so what you see here in your packet is a diagram of a hair cell. Okay, so the purple part is the actual hair cell itself. And then these long projections are not just cilia, but we actually call them stereocilia because the way they move, the direction that they move, kind of like a stereo, is going to determine what is happening. The yellow portion underneath the hair cell is actually a sensory neuron. And the pink part surrounding the hair cell would be a supporting cell. They are there to help with the functioning of the hair cell. So as you can see, the stereocilia have different lengths. So if the cilia move in this direction and it's a gentle force, then maybe only the longest of the stereocilia will move. If it's a stronger force, then some of the shorter one would also move. And that's actually what helps to determine kind of the strength of the stimulus. If the cilia move to the left, that results in stimulation. Whereas if they move to the right, it is inhibition. So what this does is it simply determines which type of neurotransmitter gets released by the hair cell itself. Now, we're going to start by taking a look at equilibrium. We have two types of equilibrium. We have dynamic equilibrium, which provides our balance while we move. And we have static equilibrium, which is responsible for our posture and stability when we are not moving. Now, each of these is monitored within the inner ear, but in slightly different areas. So let's start with dynamic equilibrium. These are monitored within the semicircular canals. Now, we have three semicircular canals, the anterior one, the posterior one, and the lateral one. And you can actually take a look at those on my three-dimensional model of the ear that I have in the classroom. Each of them are positioned basically along the x-axis, the y-axis, and the z-axis in space, because that provides monitoring of each of the three-dimensional planes that we have in the world that we live in. At the base of each of the semicircular canals is a slightly enlarged region. It widens a little bit, and that is called the ampulla. Inside of the ampulla area are the hair cells, and the stereocilia of those hair cells are actually embedded in a gelatin, or almost like a jello-like substance. So this combination of the stereocilia and the gelatin is called the cupula. Now, within the semicircular canals is a fluid called endolymph. And so as our head moves, the endolymph moves, and that pushes the cupula, which stimulates the hair cells. When the hair cells get stimulated, it's going to send a message from each of the three planes to the vestibular nuclei. Now, this brings up an interesting concept of why we get dizzy. What happens is, if you think about moving fluid, when you stop swirling, okay, you're rotating, is when you get dizzy, the fluid doesn't quite stop. So what happens is you actually get continued stimulation because the fluid still is moving in those semicircular canals. And until that fluid slows down, you remain dizzy. Now, the same thing happens with, well, not the same thing, but similar stimulation will happen. It's accidental with vertigo. You actually get stimulation of those cupulas even though the endolymph is not moving. So for whatever reason, people with vertigo think that they are being dizzy, that they have that rotational movement, but they really don't. Now, static equilibrium is monitored in a slightly different location. It is monitored in the vestibule. Basically, the hair cells are clustered in two sacs that are at right angles or orthogonal to each other, the saccule and the utricle. Again, the stereocilia are embedded in a gelatin mass, but on top of the gelatin layer are some calcium carbonate crystals. Calcium carbonate is basically like seashells, and those crystals sit on top of the gelatin. Now, the combination of the gelatin area plus the crystals is referred to as the otolith. Now, what happens is, as we move relative to gravity, the crystals are going to be the part that tend to follow the gravity, and that actually pulls and moves the otolith relative to gravity, and that lets us know how we are oriented relative to that. So in other words, this is one of the reasons why when you wake up in the morning, you actually know you're laying down and you're not standing up. So this movement stimulates our hair cells. So this actually happens with linear movement as well, like in a car, as well as an elevator. When we're standing upright, it also stimulates by pressing down on the hair cells. So actually the act of the crystals pushing down on the stereocilia also tells us something about how we are oriented. So we get more stimulation the more that the cilia is distorted, and what this information does is we're allowed to process our relative position relative to gravity. So again, this is processed in the same place as our dynamic equilibrium in the vestibular nuclei. And you can see here in this diagram, okay, the otolith is in your inner ear, so gravity pushes down on those crystals, stimulating the hair cells when we're in neutral position. If we do the simple act of tilting our chin up, okay, gravity is still going to pull down the crystals towards the earth, and that distorts them. And what that will do is give us different information in terms of how we are oriented in space. So those are the two different types of equilibrium that we have. Now, hearing, the process of hearing takes place in a different location in the inner ear, in the cochlea. It uses the hair cells again, but the mechanism is a little bit different. The hair cells are shielded from all stimuli except for sound. Now, when we talk about sound, we often talk about something called pitch or frequency, and that's determined by the part of the cochlear duct that is stimulated, whereas the intensity or the volume is determined by how many cells are stimulated. So where the hair cells are located is in a location called the cochlear duct. It lies between two fluid-filled chambers. These two fluid-filled chambers are called the vestibular duct and the tympanic duct. Now, within the cochlear duct, the hair cells are located in a region that we call the organ of Corti. The organ of Corti sits above a basilar or basement membrane, which is at the upper edge of the tympanic duct. Now, the stereocilia are actually in contact with an overlying membrane called the tectorial membrane. The movement of the basilar membrane actually pushes the cilia up into the tectorial membrane and distorts them. So in this case, the hair cells themselves actually do move in response to the sound waves. So let's take a look at this area and kind of orient ourselves in terms of what we are discussing here. So you can see what we have is we've taken the cochlea and actually done a cross-section of the tubes. So this section here is actually like one of the rings on the snail shell. And you can see that we actually have three separate tubes here. This top one is the vestibular duct. The middle one is the cochlear duct. Now the cochlear duct is filled with air, whereas the vestibular duct and the bottom one, the tympanic duct, are filled with fluid. Within the cochlear duct is a square region here, which is the organ of Corti. So now what we want to do is actually take a look at the organ of Corti and see how it is set up. Get my picture up, there we go. So this is a zoomed in view of the organ of Corti. Right here is the basilar membrane, and that is what it sits on. So underneath the basilar membrane would be the fluid within the tympanic duct. You can see here that we have the hair cells, and the blue ones around them are their supporting cells. The yellow things leading away from the hair cells are the nerves, so they bundle together to form a nerve fiber. And laying over the top of the hair cells is the tectorial membrane. Okay, so this is the basic setup here for the cochlea. So now let's actually talk about how the process of hearing actually works. Okay, so we're going to go through the hearing process. The first step is that the sound waves arrive at the tympanic membrane. And when they arrive at the tympanic membrane, they make the tympanic membrane actually vibrate. And that causes, as kind of a chain reaction, the malleus, the incus, and the stapes to actually move. If you remember, the stapes is connected to the oval window, so as it moves out back and forth, the oval window moves with it, and that creates waves of fluid within the vestibular duct. Now these waves move across the cochlear duct to the tympanic duct, and as they move into the tympanic duct, they distort the basilar membrane. The vibrating basilar membrane actually pushes up the hair cells into their overlying tectorial membrane, which distorts the stereocilia. This causes the hair cells to release their neurotransmitter and create an action potential or stimulation in the sensory neurons. So the impulse is then relayed to the medulla oblongata, and it gets distributed to other centers in the brain for processing. So if we have here, let me see if I can find a better color. There we go. If we have the oval window here with the stapes attached, okay, it's going to move in and out this way. And it's attached, I'm just going to write it here, VD for vestibular duct. Underneath that, remember, is your cochlear duct, and I'm enlarging this just to make it easier to show. And then underneath that is the tympanic duct. Now the tympanic duct eventually is connected to the round window. Okay, so what happens is we get waves in the vestibular duct, and they actually start to turn, and they come across the cochlear duct. When they come across right there, that's your basilar membrane. As they come across, this starts to move up and down, which pushes the hair cells up into the tectorial membrane, stimulates the nerve impulse, or creates the nerve impulse, which gets sent to the brain. Now the waves, after they come across, they continue their turn, and then they come out, and actually the pressure is released at the round window. Now, in terms of the cochlea itself, you can see here, we actually tend to have the high frequency sounds out towards the outer edge of the cochlea, whereas low frequency sounds are detected towards the inner part of the cochlea. And if you think about the aging process, we actually tend to lose our high frequency sounds first. And it is a little bit based on location. Because those high frequency sounds are located towards the outside portion of the cochlea, that is why we lose them. So because deafness is a major illness associated with our hearing, let's talk about the couple of different types of deafness. We have conductive deafness. This is where we get vibrations that cannot be transferred from the tympanic membrane to the oval window. So sometimes we are having problems with either the membrane or the auditory ossicles, or sometimes both of them. The good thing about this type of deafness is we have many different treatment options. Doctors have a lot depending on the specific circumstances, and obviously some of that is going to depend on which part is actually causing the problem with that transfer. The type of deafness you don't want to get is nerve deafness. And this is the type of deafness that is usually associated with listening to loud music. For instance, I know Pete Townsend from The Who is almost completely nerve deaf as a result of his guitar playing with the loudness over many, many years. Okay, this is a problem within the cochlea or perhaps the nerve pathway. It can be produced by very loud sounds. What happens is the loud sounds actually can break the stereocilia off of the hair cells, and that stereocilia does not grow back. So it's a cumulative effect. The more loud sounds that you're exposed to, the more stereocilia you break off, which means the less stimulation you get with those particular sounds. After a while, you break off enough stereocilia that you can't hear that particular pitch at all. The problem with the nerve deafness is there are very few treatment options for this. So there's really not a lot that doctors are able to do for people with nerve deafness. Okay, so that kind of concludes our look at the senses and all of their associated diagrams.